Paul Thomas, Senior Editor with Pharmaceutical Manufacturing Magazine and PharmaQBD.com. And I'm here today with John Waraska. John, thanks for being with us today.
Paul, thanks for coming by.
And John is the Director of Strategic Marketing for Life Sciences for ESA Biosciences. And John is here mainly to talk to us today about the Corona Ultra detector, which is used for UHPLC applications.
That's correct. We're just introducing it here for the first time.
And let's get right into it. Tell us about it and tell us what the key features are.
Well, it really is a proven technology now that was introduced about 4 years ago with our Corona CAD detector. And what we've done now is to make the technology compatible with UHPLC. And at the same time, we've made the technology more reliable for the production floor for QC and QA.
And a lot of development labs have been kind of going in the direction of UHPLC. And one of the reasons for that is sensitivity. And how does this kind of enable that and go in line with that?
Well, part of the whole idea of the sensitivity is the ability to see things that you couldn't see before. With a lot of the new initiatives in the pharma industry driven by the FDA, there's a desire to see more things. So the Corona addresses that in two ways. One, it's a universal or near universal detector. We see anything that's non-volatile. So you can see compounds in a preparation that you normally would miss with other technologies. The second thing is, because of the work we've done to make it compatible with UHPLC, we have sharper peaks. So this has increased the sensitivity of the detector. Where the Corona CAD had a detection limit of around 10 nanograms on column, we've been able to achieve about 250 picograms on column with the Corona Ultra. So this gives you a little more assurance that you're seeing things that are even there in lower levels. And
just to follow up with what you were talking about before in terms of universality, you're seeing more things, or you're able to detect more.
And that becomes critical when you don't know what you're looking for in a sample as it degrades or as it's metabolized.
Sure. And what about speed? Obviously, speed is important for a lot of your customers as well.
Well, that's the main driving force behind UHPLC. There are two aspects of UHPLC. One is that you can do things with the same resolution as you did with HPLC, only do it faster. And the other is you can get more resolution in the same time. The drive for speed is obvious in that it allows you to do more analyses in less time. But there's also a secondary effect in that it allows you to use less solvent. So it's a greener technology in itself. Typically, we can do things about 5 to 10 times faster with UHPLC in this detector than we were able to do with HPLC.
And you may have already answered some of this question, but what can the Corona Ultra do that perhaps other detectors can't do, especially maybe those based on UV technology?
Well, one of the problems, for example, again, in the drug industry, is when things are metabolized or when they break down, they lose the UV chromophore. So you can't see them at all on a UV detector. And then some molecules just plain have no good UV response, and you miss them with a UV detector. With the Corona, because its response is independent of the structure, we see everything that's in the sample. And that can be a great aid, particularly when you don't know what you're looking for, but also when you have products that are not UV absorbing.
And do you think there might be a lot of clients out there that maybe don't know about these capabilities or maybe haven't seen this before?
Well, despite the fact we like to think that everyone knows about the Corona, it is a relatively new technology. As I said, we introduced it about 4 years ago, but really it's the first new detection technology to hit HPLC in about the last 20 years. So, there are still plenty of people that don't know about it.
And what kind of compounds would they be using this with in general?
Mostly in the pharma industry, we're seeing a number of different things. APIs in particular, where they're looking at the drug compound, and possibly it's impurities or breakdown products. We also have a methodology using the detector where we can look at counter ions in the preparation, and we can do that at the same time we look at the API, which saves time, saves having to switch systems over to go to anions and cations. Mm-hmm. In some of the other areas, such as cleaning validation, you often don't even know what's in the cleaning agent. With this detector, you can see all those components. Mm-hmm. And then in doing mass balance work, such as in degradation studies, because of the consistency of the response, we can use the detector even though we don't know what the materials, the degradation products are. We can get a good estimate of how much is there.
Mm-hmm. Mm-hmm. And this could be used in the biopharmaceutical arena as well, large molecules, small molecules, anything?
One of the unique things about the detector is it can detect just about anything. We've gone from molecules such as glucose and lipids up to proteins. Everything responds about the same way with the technology because it really is independent of the chemical structure. Mm-hmm. So it does have a home in the biopharma area as well.
Mm-hmm. Great. Well, John Waraska, thanks so much for being with us today. Really appreciate it, and good luck to you.
Well, thank you very much for coming by.
Okay.